Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe my channel and also please give your valuable comments. In the last few classes we were discussing about the fertilization. The fertilization part is almost all over, and today onwards we will start with the fetal development. So fetal development should be studied because we know that zygote is get converted into embryo, and embryo is get converted into the fetus and then the baby. So like this there are different stages are there. So today we will start with the basic part, that is development of the zygote. We know that zygote is formed, and once the zygote is formed, thereafter how the development proceeds, so that we will try to understand. So now what happens to the fertilized ovum is that it is a unicellular embryo. Now what happens to a unicellular embryo, which is nothing but the fertilized ovum? So we know that the fertilized ovum, so that fertilization takes place in the ampulla part. So that will take place in the ampulla part of the fallopian tube, and as soon as the sperm enter inside, and as soon as there is a pronucleus of the sperm and as well as pronucleus of the ovum, when this combination takes place, then the metabolic activation occurs. So as soon as this metabolic activation occurs, the changes starts in the ovum, okay, and it will undergo the different stages of development. Suppose if there is no sperm coming and if there is no metabolic activity creation, so then usually that ovum will undergo apoptosis. Apoptosis means that particular the whatever the combination of these two, if there is in the absence of the metabolic activities, what happens? It will undergo apoptosis. Apoptosis means suicide of the cell. The cell will be get destroyed. But fortunately, when there is a fertilization, this do not occurs. When the sperm enters, this do not occurs. So what is the first step? The initial step is the fertilized ovum undergo the cleavage. Okay, this is the first step. Fertilized ovum. Fertilized ovum means it contains the chromatids of the male partner and as well as female partner. So it undergoes a cleavage. As we told in the last class itself, there is a double copy of the DNA material. The chromosome will be doubled. Paternal and maternal both will be doubled, and then they are arranged in a spindle shape. So there will be longitudinal split in the chromatids, and it moves towards the opposite pole, and the cell develop a deep slit in between, and as a result the cell division occurs. And this cell now contains a diploid number of chromosomes, 23 pairs of the chromosome. Now each part get 23 chromosomes of both paternal and as well as maternal chromosomes, and now this is called as first cleavage. Okay, so this is called as first cleavage. When the zygote get split into two, that is called as the first cleavage. And this first cleavage is usually occur site just proximal to ampulla, because proximal in the ampulla itself the fertilization takes place. The first cleavage also takes place in the ampulla itself, just proximal to ampulla. So this happens, fertilization occurs in the day 1, suppose. On the day 2, the first cleavage occurs. That means almost all after 30 hours. After 30 hours, usually 24 to 30 hours, the first cleavage occurs. Okay. Now, there will be second cleavage followed by this. So this will be followed by the second cleavage, and the second cleavage will occur by 2 days. Okay. Second cleavage will occur 2 days. After 2 days. So we have got 2 cells. 2 cells further divides. Now we have got 4 cells. So formation of this 4th cell will occur on day number 2 of fertilization. Then there will be third cleavage. So third cleavage, we will have production of almost 8 cells. Okay, so we have got 8 cells on the third day. Okay, so all these things are happening when it is moving towards the uterus in the fallopian tube. That means still it is there in the ampulla. From the ampulla, first day onwards, after the first cleavage, it start moving towards the uterus. So second day, third day, so slowly it is moving towards the uterus. Okay, so this 8 cell structure, so that is slowly moving into the uterus in the fallopian tube. So now what happens in the first week? Later, once there is 8 cells are formed, the 8 cells are further divided into 16 cell. Okay, so later there will be formation of a 16 cell stage, and this 16 cell stage that looks like a mulberry, and therefore it is called as early morula stage. It is called as early morula stage. So this formation of the morula occurs around third day of fertilization. Okay, so still it is not into the uterus, now still it is in the fallopian tube itself. Now around completion of the third day, after the completion of the third day, usually this particular morula, early morula, so that will fall inside the uterine cavity. It is now, so far it is slowly traveling in the fallopian tube from the ampulla towards the uterine tube, uterus. Now usually after third day it will fall on the uterine cavity. Now in the morula stage, what actually happens is we have got our 16 cells, isn't it? The inner cells, they talk together intimately, just like in a cricket match and all. When a wicket falls, all the members, they come together and they grouped, isn't it? So there will be layer, first layer, second layer, third layer, like this, even in the football players also. Okay, so just like now we have got 16 cells. 16 cells are now arranged, the first layer which contains 3 cells, second layer which contains 4 cells, third layer which contains 8 cells, like this. So they arrange, and they become compact. Okay, they become compact. So they become compact, and this is called as a process of compaction. You can see in the picture, this is called as process of compaction, where the cells are arranged in rows. Okay, in rows. Now once the compaction occur, then they develop some electrical stimulus, and the extensive gap junctions will develop in between the cells. There will be extensive gap junctions will develop in between the junctions, in between the cells, and the metabolic signals and ions now will go each other's. Now there is a transportation of the metabolic signals and as well as some of the ions. So these cells will go to develop the baby. That means the innermost layer which are present in this particular circle, the innermost layer which are present, okay, so they will go into develop the baby in future, and it is called as embryoblast. The outermost cells which are present in this compaction, okay, outermost cells which are present, so they become the outer cover of the baby. They become the outer cover of the baby. Therefore inner cavity, inner mass of the cells, it is called as embryoblast, and the outermost part cover, that is called as a trophoblast. That is called as trophoblast. So that particular innermost cell mass, that will go into develop embryo and later fetus in future, whereas the outermost trophoblast, so that will form the coverings and protective layers for the future, for the fetus in future. Now the morula which is fallen into the uterine cavity, now it floats in the uterus for 1 or 2 days in the uterine fluid. Okay, that means almost at the end of the third day to fall on the uterine cavity. Fourth day and fifth day, it is just floating in the uterus, in the uterine fluid. Now what happens in this morula stage? The outermost cover is zona pellucida. Now through the zona pellucida, few amount of the fluid now goes inside the morula. Okay, through the zona pellucida, through some amount of the fluid now start move inside the zona pellucida. So it enter inside the cavity, inside the morula. Okay, so now through the zona pellucida, few amount of the fluid goes inside the morula and it start producing a cavity filled with the fluid. So because of the collection of the fluid there, the cells are pushed to side. Okay, to some extent they are pushed into the side and fluid start accumulating there, and there will be a formation of the cavity which is fluid filled. There is formation of the cavity which is nothing but fluid filled, and there are such formation of small cavities, and now these cavities become connected each other to produce a cyst-like structure, okay, appearance, along with the rapidly developing cells. On the one side, the cells are rapidly developing because they are undergoing continuous divisions. Okay. Meantime, through the zona pellucida, some amount of the fluid which is present in the uterine cavity, so that will come inside and it pushes the cell and create a cavity. Previously small cavities are formed, later it is connected together to form a bigger cavity. This cavity which is produced inside the morula, okay, so on the day 5, so that is called as blastocyst. That is called as blastocyst. So you can see over here in this particular picture, see here actually this is the inner cell mass, it is pushed inside. This is the outermost part. This is the outermost part. This is nothing but zona pellucida. Okay, inside it is a trophoblast. Okay, outermost layer. Now the fate is because of entry of the fluid inside, what happens? There is a formation of a big cavity, and the outermost layer is now pushed to the sides, and innermost cell mass is here. Okay, so this is trophoblast, this is the embryoblast, and this is the blastocyst. This whole structure now it is called as a blastocyst. It is called as blastocyst or blastocyst. Okay, so if you want to make it very clear, this is a picture. Okay, this is the wall of the uterus, and this is a uterine endometrium, and the outermost cell mass, this is called as trophoblast, and this is the inner cell mass, that is called as a embryoblast, and this is the blastocyst cavity which is formed. And through this wall of the uterus, here is the endometrium. From the endometrium, the fluid passes through inside this morula stage and create this particular cavity. Okay, so what is the status of endometrium at the time of the implantation? Now, till the implantation has been not taken place, okay, there is formation of the blastocyst. Okay, how the implantation takes place, that we will see in the next class. Thank you very much, and please don't forget to subscribe my channel, and also please give